In this video, we will cover the physiology of the bitter, sweet, and umami gustatory cells. So the reason why we can rope all three of these tastes together is because they all go through a very similar physiology. The major difference between the three tastes is that the type of GPCR that they use. They each have a specific type of GPCR that they use, but in this video we're just going to treat this as like a generic setup because they all basically go through the same physiology anyway, so we're not really going to be specific about what tastant or what GPCR it is. So first things first, when it comes to tasting bitter, sweet, or umami, you need to eat something that has those specific tastants in it. And so right here is our little tastant, and the tastant is going to then circle around your mouth until it finds a receptor to bind to. And once it finds that receptor, it binds to it, activates it, and then the GPCR will associate with the subunits which are inside the cell. So the alpha subunit, which is here, this little guy in yellow here, he's going to be the most important in this story. When he gets activated by the GPCR, the alpha subunit will in turn go and activate an enzyme called phospholipase C, which you can see this right here, phospholipase C. And phospholipase C is responsible for basically cleaving PIP2. Now, PIP2 is a specialized phospholipid which hides out in the cell membranes of this gustatory cell. When phospholipase C comes into contact with PIP2, it then cleaves it into a couple of products, mainly DAG and inositol triphosphate, which is IP3. So we're not going to pay attention to DAG, but we're going to pay attention to IP3 because IP3 is what's going to contribute to the main physiology of this cell. So IP3 is then going to travel over to the smooth endoplasmic reticulum of the gustatory cell, and it's going to then cause a calcium release. So calcium is going to come out, and calcium, you should know by now that when calcium exits the smooth endoplasmic reticulum in any cell, it causes a whole host of changes. Within the bitter, sweet, and umami pathways, the calcium mainly causes the release of ATP. And we can see down here this little purple channel, ATP is being released through that due to calcium activation. So when that happens, the ATP actually acts as the neurotransmitter in this situation to affect the gustatory afferent neuron, to cause it to basically depolarize and send an action potential to the gustatory cortex of the brain so that you can know that you're tasting something that's bitter, sweet, or umami tasting. So let's go ahead and review everything that we just went through. So to start out with, the tastant binds to the GPCR. Next, the alpha subunit is then activated. Then the alpha subunit activates phospholipase C. Phospholipase C cleaves PIP2 into IP3 and DAG. And then IP3 opens the calcium channels in the smooth endoplasmic reticulum, which then causes a calcium release, which then causes the release of ATP. The ATP stimulates the afferent gustatory nerve, which then allows you to taste.